Sí.
Silencio, por favor. Vamos a dar comienzo. Silencio, por favor. Vamos a dar comienzo a la mesa redonda y tenemos en ella los 4 ponentes de la mañana que nos han ilustrado con pues todas sus investigaciones científicas. Y la verdad es que pues han aportado mucha luz al futuro del tratamiento del síndrome de Rett y de la mejora de la calidad de vida de las niñas Rett. Le rogaría que levantasen la mano todas esas personas que quieran hacer preguntas y ya las vamos llamando y les van aportando el micrófono. Ya pueden formalizar preguntas.
This is for the Dr. Alan. When you told that the scoliosis stopped after the regla, menstruation. Vale. Que la escoliosis para después de la menstruación en un alto índice de niñas. Quería que me lo aclarara un poco, porque me quedó así como en el aire.
So when scoliosis tends to progress steadily, but when a girl goes through menstruation, the progression tends to stop. It doesn't get worse. So let's say, and the curvature of the spine is measured in degrees. So let's say at age 13, the degree of curvature is 35. If the girl is in puberty and menstruating, it should be stable about 35 thereafter, throughout life. It may change slightly, but it will not have the same rate of progression as before the period starts, the menstruation starts. ¿Comprende?
Sí, he notado que desde que empezó con el botón mamario hasta que le ha venido el periodo ha sido cuando ha sido más acelerado la curvatura. Pero ahora ya desde que le vino la menstruación como que se ha parado. Entonces era para que si era normal eso.
Well,
el periodo de máxima curvatura durante la...
Yes, the period of maximum curvature is before the onset of the menstruation. After that, it slows or stops.
Gracias.
Buenos días. Quería preguntar en relación al doctor Alan también, en relación con la pregunta que ha hecho la compañera, si en un niño Rett hay algún momento en el que se va frenando también la escoliosis. Muchas gracias.
Yeah, I think that's a similar question. So scoliosis tends to progress until menstruation occurs, and ordinarily, on average, that's about age 13. It may range from age 11 to age 16, depending on the onset of the menstruation. That is the key feature. In a male.
Sí.
Yeah. They don't menstruate. But the issue is very interesting in males. So you have a range of mutations in males. And some of the males have very, very severe disease and rarely survive beyond age 2 or 3. So you rarely see, you can see scoliosis, but you don't see it progressing to the degree that occurs in girls. On the other hand, there are males that do much better than that. And in fact, we have a boy, a young man now, who's in his early 20s, who has no scoliosis, who speaks a few words, has a mutation. He's not normal, but he does not have any scoliosis. So I think the male story is quite different. However, you can see Rett syndrome in boys under 2 principal conditions. And there are not enough of these boys really to know. But if a boy has another mutation, it's called Klinefelter syndrome, where they have an extra X chromosome. So they're 47XXY as opposed to 46XY. And that extra X chromosome makes them, in chromosomal terms, like a female. So part of their cells are functioning under the normal X chromosome, partly under the abnormal X. I have never personally seen one of these children. There is one in the study that was seen by one of my colleagues. And I'm not aware that he has scoliosis. You can see it under another circumstance, and that's when there is a phenomenon occurs probably early after conception. It's called somatic mosaicism. So the child has 2 populations of cells, means he has, again, like a female, he could have an X chromosome which is normal, and the other population maybe has the X chromosome that is abnormal. And I have never seen one of those. We'd have none in our study. They are described in the literature, but I have no knowledge on their clinical picture.
Okay, I'm going to talk from the scientific point of view. And I think that we have a very interesting session. We had a very impressive starting lecture by Stuart, where he showed us that we can basically delete MECP2 from all the peripheral cells, and the mice seem to be very happy. However, the last lecture from E seems to paint a much more complicated picture. It seems that indeed it's not as simple as that. So my question is there a middle ground in there?
Yeah. So I guess the point that was made is that the mice are a milder model, so it could be that very subtle things you don't pick up. We do pick up some peripheral phenotypes that are genuinely due to lack of MECP2 outside the nervous system, and the one that's the most striking is actually the bone. So Rett syndrome girls are more prone to low impact fractures, and we see that in the mice when MECP2 is lacking in the periphery. And another feature we see is exercise fatigue in the peripheral knockout. But we didn't see any cardiac phenotype in the mice. We did look, we didn't see it in the peripheral knockout, but it could just be that the mice has some sort of compensatory mechanism.
Yes, that in this regard, I was talking in Germany about 4 years, and someone who was not Okay, so just that 4 years ago, I was at a meeting in Germany, and some people who were not working at all with Rett syndrome, they were actually trying to understand what were the mechanisms of heart stroke. So what they did is they induced heart stroke in rats. And then they did an RNA-seq to figure out which was the molecule that was most involved in there. And those people who had never been interested in Rett syndrome found that MECP2 was the first protein. So to me, it seems that, that fits well with the cardiac arrest. So to me, it seems that the brain may be important, but other reasons may be important. Maybe it requires a longer follow-up. I don't know.
Yes, so I guess the point I was trying to make is that the brain is the primary target, but there's no question that if you can address targets in the periphery, that may have beneficial effects as well. But, the dominant phenotypes are neurological. That's what I would suggest.
Oh, I just wanted to add one thing. I mean, nowadays we're actually looking at the brain-body, the mind-body connections. We found that it is much more connected than we initially would recognize. So basically, that's why I think it's giving us some hope. So if we can somehow enhance the peripheral functions, it might actually help the brain as well. So basically, I think we should be more open-minded, not only just focusing on something that's very difficult to intervene, but in the periphery, there are a lot of things you can do. You can maybe enhance. There are remedies you can do to prevent bone fractures. There are remedies you can do to enhance muscle functions. And then I mentioned a little bit about the cell therapy. That is also perhaps primarily work on the periphery, but it might actually help the overall conditions as well. We should just be open-minded at this point.
La doctora allí ha dicho en un momento determinado, o al menos eso he entendido, que había encontrado distintas mutaciones en distintos tejidos. Pero eso no me ha quedado claro. Esto podría decir que, cuando el mismo individuo tiene una mutación, ¿la tiene en todos los tejidos? ¿O puede tener, está hablando de distintos individuos? Es decir, ¿la mutación se generaliza en todos los tejidos? ¿O qué significa esto de distintas mutaciones en distintos tejidos?
Yeah, so even for the same individual will carry different mutations, just like what Alan was mentioning about these boys. They only have one X chromosome, but a group of the cells has the wild type X chromosome, but another group has the mutant X chromosome. So basically, because we are targeting the MECP2 locus really on during development in one cell stage embryo, but the mutation doesn't occur just all at once, and everything is the same afterwards. So the mutations occur along the way when cells become two cells. That locus is still being targeted. So there will be additional mutations being generated, or maybe in the two cells, one of them is not targeted, but later on, when the two cells become four cells, they'll be retargeted. So this targeting event is occurring as long as our plasmid is existing in the embryo. So we're creating a lot of different mutations, so which is different from, genetically, it's different from the Rett patient. But what we're trying to do is using a genetic mosaicism to mimic the epigenetic mosaicism in the patient. So the bottom line is we're creating mutant cells, all kinds of mutant cells, basically dysfunctional cells that they interact with the wild type cells together in these monkeys, and they're creating phenotypes. I don't know if I'm making it clear. Thank you.
Gracias.
So hi, my name is Mariam. I'm also from a scientific background. So I would like to have. I have two questions. So I'm gonna for different speakers today. Thank you to everybody. I really learned a lot in this session. So first was my question is for Dr. Sun, right? It's similar. Actually, it was the same question that he was having. How is this similar to what happened in human? I'm talking about the mosaicism.
So the bottom line is that we are creating mutant cells. We're creating mutant cells through genetic means. Okay, so some of the mutations will be nonsense mutations. So you basically, when we did the Western blot, we found the mutant tissues have less MECP2 than the wild type. So that by itself is
so regardless
creating deficiency.
the origin of the,
yes.
okay.
But the genetics is very different.
Yeah, okay.
Yeah, but maybe the next generation, when these moms are having babies, then we can then reselect the genetically homo.
So that was, exactly, so that was my next question. When would you expect the next generation?
So our oldest animal right now is 4 years old. In a year or so, I don't know if there's going to be a delay in sexual maturation. Basically, in a couple of years, when the eggs start to mature, then we can start to do superovulations, do IVF, and then we can select.
At this
mutants.
time, they will be homogeneous in the type of
yes,
mutation.
By then, yes.
We will be similar to what happened in humans.
Exactly, yes.
Okay. I have another question for you as well. You show different mutation rates in different tissues. Can you explain why a little bit?
Oh, I think that has something to do with because our targeting event is not occurring one time in the developing embryo. So when this one cell, first we got the construct, and it probably didn't get targeted because they develop into two. Then different construct goes to different, different portions of the construct that goes to different two cells, and then started targeting over there, or maybe even later when the cells turn into 4 cells. So basically, throughout the whole early embryonic development until perhaps the blastocyst, we will have these targeting events independently happening in different cells, and then creating all kinds of mutations, and God knows, maybe even after blastocyst. As long as our plasmids are still in the cells, they will do their work.
They will act.
Yeah.
Okay.
So.
And my next question is for Nicoletta. So you mentioned almost at the end of your talk that one of your markers of euchromatin will be 5-HMC. And so you show different levels, but some significant in your mutant mice. I was wondering, is it possible that this is due to changes in gene expression only and not in chromatin organization? I'm speaking like, it's not huge, the change that you show, and 5-HMC is also related to levels of gene expression. The more that you express the gene, the most likely you will have more HMC. And since you don't have huge changes, this might mean only that some of the genes that MECP2 needs to be expressed are just changes the level of HMC because they are less expressed or more expressed. So have you ever done another type of marker of euchromatin to confirm that?
So the answer for the time being, no, we have not done anything for the time being. So we are using HMC just as a probe for chromatin accessibility, okay? Because this is what literature says, that it is a marker of chromatin accessibility. And what we are doing right now, and this is the reason why we didn't send out yet the paper, but I'm not sure we will include this data, but in theory, what we are expecting is that our animals will have a little bit more transcription with respect to the null animals or the wild type. So one of the ways we can test this, but it's not easy at all, will be to perform. You know that the RNA is very technical, but anyway, the RNA polymerase is phosphorylated while it's transcribing. So what we are trying to do is to purify the same number of nuclei from neurons of the wild type null and knock-in animals, and then compare the level of RNA polymerase phosphorylation with respect to the total RNA polymerase. And similarly, from the same purified nuclei, we want to see if we can get a statistically significant, slighter more amount of transcriptions, RNA, whatever RNA is, from the null or the knock-in. So if there are differences, this is what we're doing right now.
Okay. Thank you.
You're welcome.
Bueno, agradecer a los 4 ponentes sus presentaciones y tengo 2 preguntas para 2 ponentes diferentes. Comienzo con la doctora Jif Sum y preguntar si hay alguna previsión para la aplicación de terapia génica en monos mutados.
Okay, this is gene therapy. This is my first time to heard that actually, Monica, they're actually proceeding with the gene therapy. To me, that maybe this is a little premature because I think, for different. So one of the other things that we have found, where I didn't present, is that MECP2 mutation in different brain regions will do different things. Okay, so basically, and then also MECP2 overexpression, like if you have too much MECP2, the neurons are also not normal. So I think we are somewhat desperate because we wanted to do something to help. So now we're lacking MECP2, and can we just give some MECP2 back? But the thing is, it's a very complex circuitry issues. I don't think we know, or there's evidence that tells us that just by general elevating MECP2 levels, as I also mentioned, there are some neurons never express MECP2. So if you express MECP2 in them, are you actually causing some abnormal ectopic event? I think all these questions are not really answered yet. So I would be a little bit cautious in terms of gene therapies and where to go. Do you want it to go certain brains regions to do the gene therapy, or do you want it to, generally elevating anything? Right now, I personally, I would be a little bit reluctant to do that. So I think that since there are the monkey models available, and then there's also the wild type monkeys, maybe this is time to really in detail analyzing, because then you can use viruses. So you don't have to wait till the monkeys to, from embryos to all the way to the adulthood. You can just take different aged monkeys and maybe go into different regions to intervene with the MECP2 levels up and down, and then watch for phenotypes. And then see, and then that's why I was asking the other day, what are the parents most care about? What kind of phenotype you wanted to fix first? Because that will determine where you go in the brain. I don't think just overall elevating MECP2, maybe you didn't fix the first problem, you're creating the next problem. That will be kind of sad. But that's just my personal understanding.
Gracias. La siguiente pregunta es para el doctor Alan Percy. Bueno, yo tengo una niña con síndrome de Rett, cumplirá en agosto 5 años. Y bueno, es una pregunta un poco lanzada al viento porque sé que sin que veas el caso en particular, pues es difícil opinar, pero bueno, la tengo que lanzar. Ella nunca aparentemente ha tenido ni convulsiones ni crisis epilépticas. Sin embargo, su electroencefalograma pues, marca un foco dañado, en fin, es un electroencefalograma como diríamos, no bueno, no correcto. ¿La medicarías con algún antiepiléptico en un caso así? Las niñas con síndrome de Rett pues, tienen un electroencefalograma patológico, vamos a decir, en general. Pero si tú no has visto ninguna evidencia de esas convulsiones o ataques epilépticos, ¿es viable ¿es recomendado medicarlas o no?
That's a terrific question. And I don't think that you have necessarily two neurologists who would give the same answer. But so just to give background information for everyone, the EEG, electroencephalogram, in girls with Rett syndrome is generally normal through 2 years of age. And then about 2 years of age, there is a change. And that is the time when the EEG ordinarily or normally achieves a more or less adult picture. And so the first change in the EEG is a slowing of the background activity. And what that means is that the EEG has different waveforms. Some are pretty fast. Those are alpha waves, and those are the normal alert waves, and they're different degrees. And the slowing means that in Rett syndrome, the slowing means that the alpha activity is decreased, or in fact may even be absent. So that's the first period. That is nothing that we really have any treatment for at the present time. As far as the epileptiform changes, those can begin at the same age, and they may be random spike activities, or they may be bursts of spike activities. And in fact, if you do the EEG during the time the child is asleep, the EEG for EEG experts is definitely frightening, because the spike activity increases during sleep. And so many of these folks encourage the use of anti-epileptic medication. I personally, and I've always felt this way, that seizure medication should be used to treat clinical seizures. You shouldn't be treating the EEG, you should be treating the child. So if the child is having a seizure, yes, they should be treated. If there are no seizure activity, then I don't see any reason to use medication. We have no evidence that spike and wave activity leads to injury of the brain. Now, there are people who may disagree with that, but I think the majority of neurologists and child neurologists think that epileptiform activity may occur in anyone. Could occur in me, but it doesn't mean that I would generate a seizure, nor does it mean I should be on medication. There is one type of epilepsy which is very unusual, not ordinarily seen in Rett syndrome, which is called absence, or spike 3 per second spike and wave epilepsy, and that should be treated, and that can be eliminated. But otherwise, epileptiform activity in the absence of seizures, in my view, should not be treated.
Hola, yo quería añadir también algo en el mismo sentido a Yolanda. Es una pregunta, una curiosidad como madre, o sea que me tienen que perdonar si lo que digo es una estupidez, porque aquí en España es muy dado a medicar a estas niñas simplemente por ese electro, ¿no? Lo que estaba comentando ella. Yo, por lo que veo, mi hija está en la misma situación. La han medicado y yo me gustaría que se lo retiraran. Pero por una razón que yo veo, y quizás estoy equivocada, yo de observar a estas niñas, igual que veo que muchas no beben, no sudan, lo poco que voy viendo de ellas es que incluso niñas que tienen una epilepsia con convulsiones, tanto si están medicadas como si no, veo que es lo mismo. Es decir, las que tenían pocas crisis tienen pocas, las que tienen muchas siguen teniendo muchas. Y mi pregunta o curiosidad es, quería comentarles si el síndrome de Rett frente a la epilepsia se comporta de una manera distinta a otras epilepsias y por eso los fármacos muchas veces no son efectivos.
First of all, I will say I'm not an epileptologist, but I think that maybe this is simple-minded, but I think a seizure is a seizure, whether it occurs in a child with Rett syndrome or a child with Angelman syndrome or a child with any other disorder, or a child who has had a trauma to the brain and developed epilepsy after that. A seizure is a seizure, and it should be treated with whatever medication is determined to be ideal. However, and the same occurs in the U.S., particularly among the younger epilepsy physicians. They will treat children they will treat the EEG and not the seizure disorder. I personally don't agree with that, but that definitely does occur. Now, you have to remember that the treatments for epilepsy involve drugs that have side effects among only with regard to themselves. It doesn't depend on you having Rett syndrome if you're going to have a side effect. These drugs have a variety of side effects. Some of them are could be actually life-threatening. There are some that infect the liver because the drugs are metabolized in the liver. So you have to be aware of the potential side effects of medication. There also are specific levels of drugs that should be used. And I have seen many girls with Rett syndrome who are on 3 or 4 different medications, not a single one in the optimal amount. That is, to me, wrong. You need to press one drug, whatever you want, to its maximum level without side effects to be certain that you are getting the best bang for your buck out of that drug. Because if you have 4 medications for, let's say, pneumonia, you wouldn't treat any one of those with a suboptimal amount of the medication. You need to use optimal amounts of medication. There are different schools of thought. I am of the school, you do not treat seizures that do not occur. You do not treat epileptiform activity on the EEG. Now, what do you do when you disagree with your physician? Well, you have to, you seem like you're a reasonably aggressive person. Talk to the physician and say, I want to stop. You can do it.
No, de hecho yo no soy así. Yo confío plenamente en el médico, pero mi duda, y para él también es el primer caso que vio de Rett, él cuando vio un electro como el de mi hija, pues yo le entiendo, enseguida ellos ponen una medicación. Yo lo único que intento ver es llegar a un acuerdo mutuo, es decir, probemos qué pasa sin medicación, porque para medicar ya habrá tiempo. Es lo que yo opino. Mi médico es bastante abierto, en ese sentido me escucha, pero mi duda era, lo primero lo que ha preguntado Yolanda, que en España, no sé si es por miedo o desconocimiento, un poco medican ante esos electros y simplemente eso, que a veces me da la sensación que la epilepsia, el síndrome de Rett va por un lado distinto. Era eso, pero vamos, muchísimas gracias, no por la contestación.
Yeah, this conversation could go on forever.
Hola, yo no quería perder la oportunidad, teniendo lo que tenemos, de, desde mi humilde opinión, querer saber un poco de los que hoy habéis expuesto todo vuestro trabajo durante tanto tiempo, ¿qué veis o con qué motivación os vais de aquí de cara a un futuro para decirnos qué nos queda hasta una meta o hasta un avituallamiento para poder respirar un poco a los papás? Os miráis entre vosotros a ver quién me contesta, me da igual.
¿Por dónde quieres que empecemos?
Si quieren decir cada uno su opinión de cara al futuro del Rett.
Empezamos con Alan y vamos siguiendo este orden.
Well, I've been involved in clinical trials with drugs, not with genes, but I've listened to discussions of people involved in this, and the process for approving a therapy is very long and very maddening. It's maddening for you, and it's maddening for me, because the regulatory agents have definite requirements that must be met. First of all, they're engaged in safety. If a drug isn't safe, and they want to prove that it is safe, but if it isn't safe, they will not consider it any longer. Once they have established a safety profile, then they can go for a pivotal trial. So the first phase is to try an agent in a, quote, normal population or control population. The second is to try the agent in a population with the disorder to look for both effects for safety and for effects for improvement. Most phase 2 trials should have as an endpoint, primary endpoint, safety. And then a multiple secondary endpoints to decide what is the favorable picture for that agent. Once that's been done, and it has to be done in the US at least in a series. The first trial is in individuals 16 and older. And once that's passed, then they can move to younger individuals. And usually they don't go below age 5. They may go below that, but usually not below age 5. Once they've achieved that, then they go to a phase 3. And in a rare disorder like Rett syndrome, they'll accept one pivotal phase 3 trial. It's a much longer trial, ordinarily involves a year. It requires a period of drug exposure for all of that year, but in a control situation, placebo, some getting no drug, some getting the agent, and in the last part of the, everybody gets the agent. And that period of treatment from control and case varies from 3 to 6 months. If the results are positive, then the company will go to the FDA and ask for approval to market the agent. That whole process takes billions of dollars and at least 5 to 10 years, depending on how early in the process the agent is. If it's a repurposed drug, it's shorter. If it's a brand new drug, it's longer. I think that the same phasing of activity is going to occur with gene trials. I know, because I know, that the FDA is very cautious of genetic trials because of 2 situations. One occurring in around 1990 in another disorder, which ended up very badly in the US. And the other trial was in, I think, France, with a disorder of immune deficiency. And the trial seemed to work, but the individuals developed cancer. Probably from the agent, not from the agent, but from the vector or whatever they use to package the gene. So the FDA is very, very cautious about gene therapy. I haven't spoken with them about that, but I think that's what the message is going to be. Caution.
Me refería a con qué ilusión o con qué motivación salís los que os dedicáis a esto de este encuentro, con las novedades o con los secretos que no se pueden contar y tal, pero que sabéis que hay entre vosotros. Como que entre vosotros sí que sabéis más cosas, pero no se pueden decir o no se deben, pero sí que os vais con más ilusión, pienso yo.
Well, yes, I'm an optimist, but I also am a realist, and I understand that these things take time. It's, as I said before, it's maddening to me that it takes so long. It must be worse for you, but it is a process that everyone must go through.
Gracias.
Yeah.
Me ha parecido entender que querías un poco ver la opinión de los 4. Entonces, yo creo que ahora le toca el turno a Stuart Cobb.
Okay. I thought Alan was the spokesperson for all of us here. Yeah, I'm staying. I think one has to be optimistic. I guess, so Rett syndrome is a very challenging disorder for the companies and organizations developing therapies. There's a lot of things that make it very difficult. But having said that, there's a lot of activity. So there are a lot of companies interested and have programs in Rett syndrome. Some of them are publicly available, so they're mentioned on websites and things, and others are not. But there is a lot of activity and interest, and I think one can be hopeful that, given that there's so much effort, that progress will be made. Yeah.
Nicoletta.
Okay, so although I totally agree with Alan, what did he say, I want just to say a few things. So I think I'm working on this disease since almost the very beginning, because I started to work on this by chance when I was doing my postdoc on chromatin structure in the States, and then it became, MACP2 became the cause of Rett syndrome. So I started to attend as a scientist, not as a clinician, a lot of meetings on Rett syndrome. And for the very first, I would say, 8 to 10 years, no one was ever speaking on any preclinical trials. There was nothing at all. And then suddenly, we started to talk about drugs, possible drugs. So I think this is an injection of optimism, okay? So I totally believe, I'm optimist as well. I think I'm realistic as well, and I tend to be very cautious with parents, because I think we need to be cautious with you, because we cannot sell dreams. So I think we are probably in the right moment for starting to obtain in future years, I cannot say how many years, drugs that will ameliorate symptoms. Will not be the cure, but will ameliorate. And this is already quite important. So in this, I'm very optimist. I think anytime I come to this meeting, apart that I'm really dedicated only to Rett syndrome, and it's my life. My son, when he was 5 years old, they were asking, what is your mama doing in terms of work? She's doing Rett syndrome. So because I'm only talking about this and so on. So it's really my dream to retire and see the solution. So this is what I'm hoping. But I think anytime we come to this meeting, we should think as scientists, because we are responsible of what is going on. And our mistake, anytime I hear the rumor or there are companies and stuff like that, on one side it's very good, it's very, very important, but on the other side it's very dangerous. What is going on, we are too much pushed, we are too much stressed, there are too many labs that don't control enough well the data. Alan Percy was on a paper that really, it was a 2010 paper, I don't know, that set the rules for preclinical trials. They were telling us how we should study animals in order to get the best. No one is still doing this. So I think we need you, because we need to find a better way for working. We are struggling for getting money, and there are some labs that don't have this problem, but all the other labs have a lot of problems, but still I think they can do good science. And therefore, because they are so much stressed out, all the labs, because they are competing for publishing and everything, they are not doing science in the best way, and we need to stop this process. It's going worse and worse and worse. And honestly, I don't like this way of working, and I don't want to teach this to my students. So I think we have to learn the science is rigorous, and we are doing science for helping someone. So it's not important to be the best, the most famous, the most rich in the lab. We really have to improve our scientific rigor. This is my opinion.
Okay, I guess I'm a little bit even more positive. Positive in a sense because in recent years I actually go back to China a lot, and then China has a long history of its Chinese traditional medicine. So it's been thousands of years practicing, and there are actually a lot of secrets hidden there. For example, I have been exposed to this medication that can be used to treat Duchenne muscular dystrophy patient, which is a genetic disorder. So you have a disease mutation, your muscles starts to become atrophy, and most of the patients die at age 30, before age 30, because of the deteriorating of the of the muscles. And then there is this medicine, apparently very safe. And it's a family descent. So in China, there are lots of these pockets. It's not like the whole Chinese traditional medicine sort of like on the book does not seem to be very rigorous. It has lacking a lot of the controls, but people have been practiced this. There are secrets still hidden in those. Right now, it's needing really good scientific methodology to basically discover those and then to study them to verify the efficacies of the practice of the medication, of the therapeutic practice, and once that is something that we can do. So we can use very rigorous scientific criteria to reevaluate those secrets that's been existing for thousands of years, and then rediscover new things that is totally unexpected. So for example, this Duchenne muscular dystrophy methodology, right now I'm working with the family descendant of, practicing the medication, and then we're basically discovering new ways. So ways that are out of the current knowledge is being telling us, like how muscle development works. So I think maybe these things can be, so like we as a scientist are also need to be thinking outside the box. It's not that everything we've learned so far, if we limit it to this, it'll be very difficult to find a cure, but maybe just complete outside the box, maybe bringing something new thinkings into this. I think I also agree with Nicoletta that we are gonna try to first develop things to make the lives better. So maybe going from the periphery to take care of a lot of the day-to-day issues, and then eventually moving into the center. So there are these some other Chinese traditional medicines that works very good on the neuromuscular regeneration. So we're looking into that as well. And then I think, and those medication has been practiced for such a long time, so that time is a huge element to tell you whether this therapy is safe or not, because it's been going on. It doesn't, maybe the scientific doctors, they don't understand the molecular mechanisms underlying that, but the timing has made this therapy survive. That is suggesting to you there is a very good safety profile behind that, and then we need to figure out how it works. And then those things, I'm just trying to dig those things out. And then I think for Rett syndrome, maybe unexpectedly we'll have some surprising good news. That's my thinking.
Yeah, very, very quick question to Dr. Stuart Cobb. So we know that too many copies of MECP2 is detrimental. Do you know if it's true with the very truncated version you've been describing?
It's probably true. So the shortest truncate version is slightly destabilized. So with one copy of the truncated version, the level of MECP2 is actually reduced. So we believe that actually these, so for gene therapy, we believe actually that a small amount of functional of MECP2 in cells can actually have quite a big effect. So one of the strategies to overcome these issues of dosage is actually to deliver vectors which deliver sort of sort of normal level of MECP2 to cells, so that even cells that are receiving 2 or 3 copies will not sort of slip over into overexpression phenotype. So certainly the short truncated one's slightly destabilized, and we're actually now trying to develop other versions that have even lower levels to test those.
Got it.
There's actually evidence in other diseases that you do not have to return the level of the element you're trying to increase to 100%. In some disorders, it could be as little as 25%.
No sé a cuál de los cuatro formular la pregunta, pero si las niñas nacen los niños nacen con la mutación, ¿cuál es la explicación de que se manifieste tan tarde? En algunos casos después de los 12 meses y teniendo una evolución normal.
that also is an excellent question. So you have a mutation, why doesn't it express immediately? And I think we go back and we at least rationalize it with our understanding of the development of the brain, or the neurobiology of the brain. So that during the first several months of life, there is a rapid evolution of brain cell maturation, so that you have a, not a scientific word, a tremendous amount of membranes formed in the brain related to nerve cells, which increase the size of the brain. But the number of cells there is there. It's not going to get more neurons, is not going to increase. So this evolution of the brain and the activity that is occurring simultaneously with that, and the elaboration of good connections between neurons, we call them synapses, so they can talk to each other. That is a phenomenon that is occurring very rapidly during this time, and we think that at the end of the first year of life, that is about the time when you should see these more advanced functions develop. So when you're a newborn baby and for the first 6 months, what does your baby do? Not very much. They eat and they sleep and they move around a little bit and they fuss. But as time goes on, they gain more functions. And it's that evolution, and they reach the critical point where the brain can't keep up with the expected development of, let's say, sitting up or crawling or walking or playing with objects, or in fact saying a few words. So it's the process of neurobiology and development.
But in some cases they talk and then they stop talking. So
this is correct.
How come?
And that depends on, in some regards, on either the type of mutation or the effect of the mutation, as to when or how much the impact of that specific mutation is on your daughter.
Mi pregunta es, sacaste una gráfica en la cual pones los tipos de mutaciones que hay. ¿Alguno de ustedes tiene cuáles son en cada tipo de mutación? ¿Qué es lo que esperaríamos o no esperaríamos? Sé que hay unas más graves, pero ¿qué podemos o dónde podemos encontrar algo para saber qué esperar en cada tipo de mutación?
So first of all, if you have your child has a given mutation, and we can only speak in general terms that mutation should have this degree of difficulty associated with it, whether it's a milder mutation or a more severe mutation. We can only speak in generalities. We cannot speak about the development of your child because, as I said, two children with exactly the same mutation may be completely different. It's when you have large numbers of individuals with the same mutation that you can make statistically significant conclusions or reach conclusions. So there are papers in this regard. I can only speak mainly for my own, but there's a paper, first author is Cobb, from 2014, I think, which talks about that. There are papers by Newell and Bebbington from 2008 in the journal Neurology that talk about the same issues. But we have to have large numbers of individuals with each mutation to make sense of how an individual with those mutations is going to develop. That's why we can focus on these 8 common mutations and the certain other mutations which are very similar that have enough numbers to reach conclusions. What number do we need? We probably optimally need about 50 individuals with the same mutation to draw conclusions as to their, the severity of that specific mutation.
Can I add one thing? But maybe it's more a question on my side on that. Although I understand what you're saying, I think we probably will never be able, or for a long time, to exactly predict what will happen, because there are 2 other features that are important, apart from the X inactivation. But all the other gene composition, what I'm meaning is that there are a few cases that have been published, but Alan can correct me, where the mother has a mutation, she's perfectly normal, and the siblings are very much affected. And this is depending because she will have some modification in other genes that still we don't know, that are protecting her for showing the phenotypes of the disease. So the problem is that we still don't know how many genes, we call them modifiers genes, how many genes communicate with MCP2 and participate to the severity, making therefore the disease more severe or less severe in the presence of the same mutation. So this will take longer, because even if we have 50 patients with the same mutation, we still have to consider this fact.
I agree with all of that, and then I'd go back to the point that I tried to make earlier when I presented that the distribution of the mutated chromosome in the brain is critical. If you have a preponderance or an abundance of abnormal chromosome in this part of the brain and this part of the brain in another child, those children are going to be completely different. Completely different. And so reaching a conclusion just knowing what your blood test showed is, I don't think it's something that anyone should be able to say they can do. It's just not a conclusion they can reach.
Let me just add one quick thing. So like with regards to different mutations, we have been working with patient cells. Fibroblasts, we turn them into iPS cells, the pluripotent progenitor cells, and then differentiate them into neurons. So in that situation, we're not really looking at the brain, we're just trying to look at neurons as functional units in a culture dish. And then we do find that different kind of mutations will give different severities with regards to the phenotype. For example, we already know that when you knock down a gene, and then you can knock down the MECP2, then you can rescue it with different mutations. And some of them will actually have some rescuing functions as compared to, not as good as a wild type, but different mutations will have different kind of functions. So your question is scientifically, you can actually try to address that with different simplified systems. But as what Alan had mentioned, that when it comes to a patient, that is another, different ballgame with, regional specificities, and also the genetic background is extremely important as well, X activation. So that's it.
Gracias.
¿Hay alguna enfermedad genética que ya esté aplicando los vectores con resultados favorables?
Yes. So, I mean, there are a lot of clinical trials just now for gene therapy in a variety of disorders, but not so many for neurological disorders. So Avex is the company that are planning to develop a gene therapy for Rett syndrome. They have one other clinical trial, which is quite advanced. So this is for a
No digo para el Rett, digo que si ya se está aplicando en otras enfermedades genéticas con resultados positivos a humanos.
Yes, so So this company of Exis have been developing a gene therapy for another disorder. It's a neurological disorder called spinal muscular atrophy. And so this is a very severe disorder in which the motor neurons control the muscles degenerate. And they have very encouraging data from that. So the gene that they deliver, the outcome measure for that is loss of muscle function. So typically by 2 years, the children are required to be on a respirator, and ultimately it's a lethal disorder. So they have children that are able to use their muscles, children that are sitting up, and even children that can walk months beyond when they would normally be on a respirator. So they have presented that data to the FDA, and the FDA has recommended that they apply for licensing. So although, as Alan was saying, the initial clinical trials are always safety trials. So really, any initial clinical study is really to see whether the molecule that you're delivering is safe. But for rare disorders especially, quite often there's an outcome measure built in, and in this case, for this gene therapy, the outcome is very encouraging, such that they're trying to fast track the licensing of that. And this is why actually this company of Exis has their stock price has soared on this, and this is causing them to invest in other disorders. There are other diseases, so that's delivered peripherally by intravenous delivery of vector. There's another disorder called GAN, which is giant axonal neuropathy, and it's the first gene therapy clinical trial in which the vector's delivered by lumbar puncture. So it's a bit like an epidural. It's a needle into the base of the spine, and that trial is ongoing at the moment. So that is probably the preferred route of administration for a gene therapy if the trial goes ahead for MECP2. So the preclinical data in monkeys for MECP2 has been by lumbar puncture so far, safety studies in primates.
Era preguntar si ya había pioneros en el uso de los vectores ya en humanos, era por eso.
Can I add only a comment that might be very naive because I'm not a gene therapist, but I was at a meeting in Israel and they were talking about this. And so there are diseases that have been fixed, but not providing the virus directly, but using the virus to affect, performing gene therapy on the cells, and then the cells have been given to the patients. This is for Adashit, that is a lethal disease. It's for also to fix epidermolysis bullosa, it's called. And so these diseases are completely curable by gene therapy, but again, it's a gene therapy that you apply to the stem cells of the patients and then you transplant the cells. And I was asking them, so there was one of these persons that has already cured many patients for fibroblast problems, skin problems, and so on through gene therapy. And I asked them, so is it dangerous? And he was telling me, and he was very confident, but I leave it to you, I just tell what I heard, is that he believes that it's really depending on the gene that we are using, if gene therapy is safe or not. So he said, I had never seen any problem because of the insertion of this gene in the genome, never ever, never cancer problem, never ever. And I will bet, and I did a lot of studies on mice, and there are no problem at all, because it claims that this gene is not dangerous when it's used in gene therapy. So maybe this is something also that we should test in animals, to test how many times, if we use gene therapy, we can produce, if we can ever, any kind of cancer or stuff like that. But this is a naive comment.
Yeah, so I guess I'd add, this. So the concept of gene therapy is contentious, and it divides clinicians, and it divides scientists, and it divides families as well. So I mean, it is a risky procedure, these initial experiments. We really don't know until we actually get into a clinical trial what the potential problems will be. You know, it offers the potential for quite a profound effect, but no one's really denying the fact that we're sort of stepping into the unknown. But what Alan was saying is that the regulators, the FDA or the European Medicines Agency, you know, their first priority is safety, so they really do everything in their power to ensure that safety is paramount in actually being allowed to launch a trial. So they will look at all the preclinical safety data and make a judgment based on that.
I wanted to add a comment with regards to gene therapy versus cell therapy versus using cell as a carrier for gene therapy. So, because Nicoletta was just mentioning, in the periphery, if you're fixing a hematopoietic kind of a system problem, it's very easy to get your hematopoietic stem cells, either fix it or you get it from a different donor to fix it. So in the case of Rett syndrome, I think that's actually a positive perspective, because the cells, you do have 2 chromosomes of X, and then if you are dealing with cells, you can totally pick and choose the ones that are good, you know, with the mutant allele being inactivated, and then you can expand those cells. So if, let's say, we can get cells, neural stem cells, for example, in the very, you know, we selected the one from the same patient, but we select the ones that are properly X inactivated, expand them, and then doing some transplantations to basically enhance the wild type cells in the brain. You're not going to get rid of the mutant cells, but you're giving more wild type cells. That is something that's actually worth to think about. So you can definitely use cell therapies for Rett patients to take care of the periphery, but with regards to the CNS, there are some explorations into, you know, putting in more good cells into the brain that might actually dampen down sort of the influence from the bad cells. That, you know, may be also something that's worth to think about.
Hola, soy padre de un varón con la deleción en el FOXG1. Quería saber, mi pregunta es, tiene 14 años, si se están realizando estudios también en el FOXG1.
What kind of studies, I mean?
Investigación, generalmente. Quiero decir, es que si las niñas son pocas, los varones son muy pocos, entonces nosotros nos sentimos como perdidos. Y quería saber si sería aplicable todos los descubrimientos que se hacen a los varones.
First of all, FOXG1 is an autosomal disorder. It's in chromosome 14. So boys and girls should be affected presumably equally. There is research going on both in the basic science level and this clinical study that we're doing. I can't say that I know all the research going on, but I know that it is going on. It is, let me say, a late arrival to the scene as opposed to Rett syndrome. So it has to go through its own evolution and maybe can take advantage of some advances that have been made with Rett syndrome or other disorders, particularly with genetic modifications, to advance the research in that disorder. It is less common or less recognized. It may be as common, but it just isn't recognized yet. And I don't think we have the answer to that question. The same is true for CDKL5, although that is an X-linked disorder. But it is, it does affect both boys and girls, and it is also a little behind the curve in terms of research efforts, and is also less common. At least the recognition is less common. So I think progress is being made, and I hope that it is accelerating and will catch up with Rett syndrome and other disorders.
Gracias, gracias.
¿Alguna pregunta más?
A comer.
O ya nos vamos a comer. Ya parece que hay apetito. Bueno, lo que sí me gustaría antes de finalizar es dar las gracias a los 4 ponentes, a Stuart, a Nicoletta, a Alan y a G. Y bueno, darles gracias sobre todo por todas sus aportaciones, porque yo creo que nos están dando muchísima luz en este camino. Yo desde luego he anotado muchas cosas y bueno, yo aparte de yo soy más optimista en este momento de lo que era cuando iniciamos este encuentro. Así es que bueno, pues muchas gracias a los 4 y reanudamos a las 3:30.